Assalamu alaikum. Today we want to talk about chapter 21, carbohydrates. And we will cover, inshallah, the following sections: an introduction to carbohydrates, handedness of carbohydrates, the D and L families of sugars, drawing sugar molecules, structure of glucose and other monosaccharides, some important monosaccharides. What are carbohydrates? Are a large class of naturally occurring polyhydroxyaldehydes or ketones. Monosaccharides or simple sugars have from 3 to 7 carbon atoms and one aldehyde or one ketone group. Now, if the sugar has an aldehyde group, it is an aldose. If it has a ketone group, the sugar is classified as a ketose. And this is called the Fisher projection formula for glucose. Glucose 6 carbons from 1 all the way to 6. Since it contains an aldehyde group, it is an aldohexose. It is a monomer for starch and cellulose, which are the major source of energy. Here we have 1, 2, 3, 4, 5 carbon sugar, an aldopentose called ribose, a component of ATP, coenzymes, and RNA. Then we have also a 6 carbon sugar called fructose, is a ketohexose, and this is present in corn syrup and fruit. Now, monosaccharides undergo a variety of structural changes and chemical reactions. They form disaccharides and polysaccharides, which are called complex carbohydrates, which are polymers of monosaccharides. Functional groups are involved in reactions with alcohols, lipids, or proteins to form biomolecules. Now, chiral compounds lack a plane of symmetry and exist as a pair of enantiomers in either a right-handed D form or a left-handed L form. If we take this simplest sugar, 3 carbon sugar called glyceraldehyde, we have this carbon is chiral. Chiral means it's surrounded by 4 different groups. As you can see here, this carbon is chiral. We look at the OH. If it is to the right, then it is designated D-glyceraldehyde, and this is D-dextro, yani right-handed. And if OH on the chiral carbon is to the left, then it is designated L-glyceraldehyde. L-levos means left-handed. So, what is the relation between D and L? They are enantiomers. Now, when we go to aldotetroses, yani 4 carbon sugars have 2 chiral carbon atoms and can exist in 4 isomeric forms. Look here, we have this erythrose. It has 2 chiral carbons, as you can see. Now again, to assign configuration for erythrose, we look at the highest number chiral carbon. We look at OH, it is to the right, then this is D. And its mirror image, OH to the left, this is L. So we have 2 non-superimposable mirror image isomers called enantiomers. The same for threose. Here you see OH to the right D and OH to the left L. By convention, the carbonyl group and the terminal CH2OH are drawn pointing to the right. In general, a compound within chiral carbon atoms has a maximum of 2 to the n possible stereoisomers and half that many pairs of enantiomers. In some cases, fewer than the maximum predicted number of stereoisomers exist because some of the molecules have symmetry planes that make them identical to their mirror images. So, for example, if we go back here, how many chiral carbons we have? In 2 to the n is the number of chiral carbons. So we have 4 stereoisomers exist for erythrose. 2 of them have a D configuration and 2 have L configuration. Again here, the D and L families of sugars, drawing sugar molecules. Now in a Fisher projection, as I told you about, this is Fisher. The aldehyde or ketone group is always placed at the top. Then you replace the chiral carbon with the intersection of 2 lines like this, and we have 4 different groups. So this is again chiral, and this is the Fisher projection formula of glyceraldehyde. Here, now notice how the horizontal line here points out of page toward you, the viewer, whereas the horizontal bonds are bonds behind you, away from you, the viewer. And the same here, dotted away and the dashed toward you, the viewer. So here the result is that OH and OH groups projecting above the page are on the left of the chiral carbons, and groups projecting behind the page are above and below the chiral carbon. Above and below. This is the chiral carbon. So the groups above and below, they project behind the page, and groups, the H and OH, that are on the left and right side of the chiral carbon project above the page toward you, the viewer. Again, monosaccharides are divided into D sugars and L sugars based on their structural relationships to glyceraldehyde. As I told you, this is the Fisher projection formula for D-glyceraldehyde. Why D? Because OH is to the right of the chiral carbon. So this is D-glyceraldehyde. If we put a mirror, its mirror images, OH to the left, this is L-glyceraldehyde. So these two are non-superimposable mirror image isomers called enantiomers. Now in the D form, the OH group on carbon 2 comes out of the plane of the paper and points to the right. In the L form, however, the OH group at carbon 2 comes out of the plane of the paper and points to the left. Then right, left, right, left. Dextro, right, levo, left. So D and L derive from the Latin dextro for right and levo for left. In Fisher projections, the D form of a monosaccharide has the hydroxyl group on the chiral carbon atom, farthest from the carbonyl group pointing toward the right, whereas the L form has the hydroxyl group on this carbon pointing toward the left. So again, if we look here, how to assign D configuration for a monosaccharide like this, two pairs of aldohexose enantiomers. Here we have allose. Now, how many chiral carbon? It has 1, 2, 3, 4. Now number 4 is the highest numbered chiral carbon. Since OH is to the right, then this is D. Its mirror image is L. Again, if we take glucose, this is the highest numbered chiral carbon. OH is to the right, it is D-glucose. OH to the left, it is L-glucose. Chirality and drugs. The goal of modern drug development is creation of a drug molecule that binds with a specific hormone, enzyme, or cellular receptor. Because most biomolecules are chiral, a chiral drug molecule, single isomer, is likely to meet the need most effectively. Now, the route to a chiral drug molecule begins with chiral reactants and an enzyme, or with chemical synthesis of a pair of enantiomers that are then separated from each other. Enantiomers can have different effects. Maybe the active ingredient in the painkiller and anti-inflammatory Aleve is sold as the single enantiomer, as a single enantiomer. The other enantiomer causes liver damage. So this shows you the importance of enantiomers. So in the synthesis of drugs, they take, for example, the D configuration. Maybe this D configuration is a painkiller, and the L enantiomer, or the L configuration, causes liver damage. So again, here it shows you the importance of drug synthesis in the correct configuration. Now, monosaccharides with 5 or 6 carbon atoms exist mainly in cyclic form when in solution. Aldehydes and ketones react reversibly with alcohols to yield hemiacetal. So this is reaction in organic chemistry. Aldehyde plus alcohol produces a hemiacetal. This is a reversible reaction. However, if the aldehyde and the hydroxyl group are on the same molecule, as in the case of D-glucose, this is the open chain form or the so-called Fischer form. Now carbon 1 reacts with carbon 5, as you can see here. We arrange them here. Now carbon 1 reacts with, now oxygen act as oxygen of this oxygen act as a nucleophile. It attacks the carbon and carbon here, rotate and then ring close. Then you get two forms. If the OH on the newly synthesized chiral carbon number 1 is up, then it is beta D-glucose. If the OH is on the anomeric hemiacetal carbon atom is down, then it is alpha. So beta OH up, alpha OH down. Now what is the relationship between this alpha and beta D-glucose? They are called anomers. Notice here, if we count how many chiral carbons in the Fischer, 1, 2, 3, 4. Now this is called the Haworth projection formula. How many here we have? In addition, now we have a newly synthesized chiral carbon. So we have 1, 2, 3, 4, and 5. We have 5 chiral carbons. So there is an additional 1 chiral carbon synthesized. It is designated carbon 1 and it's called the anomeric carbon. The OH can be down or the OH can be up, as you can see. Now D-glucose can exist as an open chain hydroxyaldehyde or a pair of cyclic hemiacetals. The cyclic forms differ only at carbon 1, where the OH group is either on the opposite side of the 6-membered ring, alpha, or on the same side, beta. Now the CH2OH group in D sugars is always above the plane of the ring. This is always above the plane of the ring. Cyclic monosaccharides that differ only in the positions of substituents at carbon 1 are known as anomers. And carbon 1 is said to be an anomeric carbon atom. So again, if we go back here, what we call beta D-glucose and alpha D-glucose, they are enantiomers. Why? Because they differ only in the configuration around anomeric carbon number 1. Here OH up, here OH down, and the other OHs are same. Now notice, when OH, the hydroxyl group, and the CH2OH, these are in cis position, then it's beta. When the CH2OH on carbon 6, and the OH are in trans relationship, then this is called H alpha. So again, beta up, cis, alpha down, trans. Although the structural difference between anomers appears small, it has enormous biological consequences. Crystalline glucose is entirely in the cyclic alpha form. In water, equilibrium is established among the open chain form and the two anomers through mutarotation. So you can alpha D-glucose and beta D-glucose, they exist in solution in this relationship, 36% alpha and 64% beta. However, this is the open chain D-glucose in very low percentage. You could convert the alpha to beta or the opposite through an enzyme called mutarotase, and this is called mutarotation. So the conversion of alpha to beta or beta to alpha is called mutarotation, and the enzyme is called mutarotase. We need an enzyme to do this. This is a summary for monosaccharide structure. You can read it by yourself. After you study all this, this is your summary. So if you know this summary, then you understood the rest. Some important monosaccharides. Monosaccharides can form multiple hydrogen bonds and are generally high melting, white crystalline solids that are soluble in water and insoluble in nonpolar solvents. Glucose مثلا. Most monosaccharides and disaccharides are sweet tasting, digestible and non-toxic. مثلا glucose and sucrose, except for glyceraldehyde and aldotriose and fructose, ketohexose. The carbohydrates of interest in human biochemistry are all aldohexoses or aldopentoses, and most are in the D family. So naturally occurring carbohydrates have D configuration, not L. So know this. Glucose is the most important simple carbohydrate in human metabolism. It is the final product of complex carbohydrate digestion and provides acetyl groups for entry into the citric acid cycle as acetyl-CoA, as we will see in coming chapters. Maintenance of an appropriate blood glucose level is essential to human health. Of course, as you know, there are two hormones that control blood glucose level, insulin and glucagon. These two, they maintain blood glucose level. Galactose, D-galactose, is widely distributed in plant gums and pectins and is a component of the disaccharide lactose. Galactose is an aldo, so this is the structure, the Haworth structure of alpha D-galactose. So as you can see, galactose is this is the open chain, is an aldohexose. Galactose is an aldohexose. It differs from glucose only in the spatial orientation of the OH group at carbon 4. 1, 2, 3, 4 here. OH, instead of up, it's down in glucose. Now in the body, galactose is converted to glucose to provide energy and is synthesized from glucose as needed. So this is the fate of galactose. It is converted to glucose to provide energy. Fructose, on the other hand, D-fructose, often called levulose or fruit sugar, occurs in honey and many fruits. It is one of the two disaccharides combined in the disaccharide sucrose. It is a ketohexose rather than an aldohexose. In solution, fructose forms 5-membered rings. So again, this is the open chain of D-fructose. لاحظوا, fructose is a keto rather than an aldo. It is a ketohexose because of keto group. Now in solution it forms a 5-membered ring, as you can see here. This is now carbon 1, 2, 3, 4, 5, and 6. And this is on carbon 2 here, the OH down, on alpha down, and beta, the OH is up. Alpha down, and this is the newly synthesized anomeric carbon, but here it takes number 2 because we started with carbon 2 here because this is keto. In sugar, this was carbon 1. So alpha and beta D-fructose are also known as anomers. Ribose and deoxyribose. Ribose and its relative 2-deoxyribose are both 5-carbon aldehyde sugars. These two sugars are most important as parts of larger biomolecules. Now ribose is constituent of coenzyme A, ATP, oxidizing and reducing agent coenzymes, and cyclic AMP. Now 2-deoxyribose differs from ribose by the absence of one oxygen atom. That is the OH group on carbon 2 here. So this is your beta D-ribose. Now if you replace the OH with hydrogen on carbon 2, it becomes beta D2-deoxyribose. Ribose in RNA, deoxyribose in DNA. Okay, finally in part 1 here, some important monosaccharides, cell surface carbohydrates, and blood type. Human blood can be classified into 4 blood group types called A, B, AB, and O, based on the presence of 3 different oligosaccharide units designated A, B, and O. Among the targets of antibodies are cell surface molecules that are not present on the individual's own cell, thus foreign blood cells. Now, type O on cell surface are similar in composition to those of types A and B. People with blood type A, B, and AB all lack antibodies to type O. cells, making individuals with type O blood universal donor. So type O blood is universal donor, can give to all other types. People with type AB, however, blood have both A and B molecules on their red blood cells. Their blood contains no antibodies to A, B, or O, and they can, if necessary, receive blood of all types. So these are called universal acceptor. Okay, this is the end of part 1, and thank you.